Okay, so hello guys. So to continue with our discussion on gram-positive bacilli, so let's proceed with the first group. We have our Corynebacterium. So the genus Corynebacterium is actually a large group of bacteria. There are about 80 or more than 80 species in the genus, but at least 50 are thought to be clinically significant. But for the purpose of our discussion, of course, we will just limit to a few. Okay, so most of the species are found as normal microbiota on the skin and mucous membranes of humans and animals alike. Now, Corynebacterium organisms can be divided into two general groups. We have the lipophilic, or those that are fastidious and fast growing and would require lipids in order for them to grow properly. And the other are the non-lipophilic group. These are those that are non-fastidious and they do not require lipids in order for them to grow successfully or luxuriantly on culture media. Okay. Now, first species of the group would be Corynebacterium diphtheriae, which is known as the causative agent of diphtheria. Mind the spelling, that's PHTH. Okay. So our Corynebacterium diphtheriae is known as the Klebsiella bacillus. Okay. To cultivate them, note that they are able to ferment glucose and maltose. They are positive for nitrate reduction and catalase, but they do not produce the enzyme urease. Okay. So disease association, your Corynebacterium diphtheriae again is the agent of diphtheria. So we have respiratory diphtheria, which is the severe form of infection. It's brought about by the presence of or the ability of the organism to produce diphtheria toxin. Now, note that Corynebacterium diphtheriae that are able to produce diphtheria toxin are actually those bacteria that has undergone lysogenic conversion. We call lysogeny, okay, or transduction, okay, yung specialized transduction. So organisms that are infected by viruses or bacteriophages may actually acquire, okay, properties of other bacteria that has been previously infected by the bacteriophage. And so these are special properties, special abilities. Production of diphtheria toxin are acquired by bacteria, Corynebacterium diphtheriae, only after they have been infected by a bacteriophage, where they were able to acquire properties, like toxin production from other bacteria. Okay. So once they have undergone lysogenic conversion, they are now able to produce diphtheria toxin. Okay. So in diphtheria toxin, there's epithelial destruction, there's inflammation. Of course, when epithelial cells are destroyed, they may die. Okay? That would be the source of necrotic tissue. Of course, our body would try to repair them, so there would be fibrin clots. Okay? And during repair, there would be a multitude of immune cells and blood cells at the same time. So the combination of necrotic tissue plus fibrin plus blood cells, that would cause the pseudomembrane formation. Okay? So if you look closely sa bunganga ng pasyente natin, look. Okay? So the tonsillitis naman. Tonsil area is inflamed, not really that swollen, but inflamed still. But look, beyond that, there is that white membrane that you see. Okay, so instead of having the entire space for air and food to enter, the space is narrowed down. Okay, so the lumen that would supposed to be your opening of your esophagus would be smaller because of the pseudomembrane formation. Okay, this would also produce the characteristic bull's neck because the neck becomes slightly swollen or enlarged. Okay. Then we have the cutaneous type of the diphtheria, where no, not spores, bacilli, no, the organism itself was able to enter through breaks on skin. Okay, so magnecrotize, so necrotic lesions on the skin. Okay, so what do we do in the laboratory? Okay, so you can work with samples from the nasopharynx, okay, especially around the inflamed area, or if you're working with cutaneous diphtheria, so you can use wound or skin samples for your specimen. Okay, so with your specimen, you can do gram staining, and then you can also perform Leffler's methylene blue stain, where the characteristic Chinese characters appearance of the organisms are more pronounced. Okay. Although you can also see the coriniform or the club shape or Chinese characters arrangement of the organisms when we do gram staining. It's just that when you use Leffler's methylene blue stain, they become more enlarged, more pronounced. Okay. Now, our organism also has the characteristic swollen appearance or the club shape appearance, technically because of the presence of metachromatic granules on ends. So this picture here, using the Leffler's methylene blue stains, allow us to see where the metachromatic granules are concentrated. So you have concentrations on both polar ends and at the center. Okay. So we call those the Babes Ernst granules. It's actually not apostrophe. Okay, there's a hyphen. Babes, no, not babe apostrophe s. Okay, I apologize for that. Okay, so babes as in whole word, no apostrophe, and then a hyphen, and then Ernst. Okay, so Babes Ernst granules ang tawag dun sa metachromatic granules nila. Okay, so cultivation can be done on uh, sheep's blood agar, where our organisms are able to demonstrate a really narrow zone of hemolysis. And then you can work with Leffler's serum, okay, or Pice agar. They usually would enhance metachromatic granule formation. That's why when you use the Leffler stain, they appear to be swollen. So it would be a good pair if you culture organisms on Leffler's serum or pie agar, and then stain them using the Leffler's methylene blue stain. Okay. You can also work with potassium tellurite medium, where they are known to produce gray to black colonies. Okay. This is also a selective media. It would inhibit growth of normal flora and other microorganisms. And then Tinsdale medium, which is a specific media made for Corynebacterium diphtheriae. Here, they produce black colonies, no, here, with halo. Parang nag-diffuse yung a color or pigment. Okay, so it's on the surrounding media. Yeah. And then there is a special test. We have the elect plate test. Okay. And then we have the modified elect test. Okay, so for in vitro. So elect test, no, the original test was used for direct inoculation, okay, animal inoculation. But in the laboratory, if you're working with plates like this, we call this the modified elect test. Okay, so what we do here is that we have a strip, no, a filter paper strip soaked in diphtheria antitoxin. So this is the actual plate. Okay, so this is our. Wait, let me find my. Okay, anyway, I can find my eyeglasses. Okay, so we have here the streaking for negative, positive control, your unknown or the specimen at the center, and then the negative control. Okay. So if you look closely, you'll see okay, precipitin bonds, okay, forming what looks to me as an X. Okay, so the precipitin bonds usually appear at 45 degrees angle between the uh, specimen and the strip. So you see here, a very light, no? Gaya nitong nasa drawing. But dito, mas magandang tingnan. Okay? Parang nag-X. Ayan. So there's a thin precipitin bond right here, and then one here, and another at the bottom part. So each corner where there's an acute angle, a right angle rather, there's a precipitin bond, that you see. So this is the positive control, and our unknown mirrors the positive control. So therefore, the unknown is positive for the diphtheria toxin. Okay. Next, we have our Listeria monocytogenes. So technically, there are about 10 species under Listeria, but only our monocytogenes is considered as an important human pathogen. So there's monocytogenes can be found uh, anywhere. They're ubiquitous. Okay, so we can find them in the environments. They have been recovered from milk, no cheese, poultry, dairy products mostly. And then I've read somewhere they can be found also in cabbages. No? So most probably they're also associated with soil, if that's the case. Remember, ang cabbages natin are low-lying plants. And so when we water them, particles of soil may enter the leaves. That's why they can be isolated from cabbage leaves. Okay, so our the treatment for this, bakit ba nasa kalagitnaan ito? The treatment for this is penicillin and erythromycin. Okay, so our monocytogenes is actually a virulent organism. It has virulence factors, no? Like adhesin proteins, like uh, fibronectin binding protein A. Okay, remember fibronectin is a surface protein or surface structures, no, or receptor sites for attachment that's found on human cells. Okay, and so these microorganisms would require something that would allow them to attach to fibronectin by proteins. Okay, so they have the fibronectin binding protein A. Okay, they have internalin. Internalin A and internalin B actually reacts with E-cadherin. And E-cadherin is also a surface protein that's used by microorganisms for attachment so that they could gain access into the cells. Okay, so fibronectin and E-cadherin are found on the surfaces of human cells, and organisms like Listeria use this so that they could attach and penetrate into the unsuspecting host. Okay. And then they have the protein P60. So the protein P60 induces phagocytosis. They're also responsible for increased adhesion and penetration of the bacteria into mammalian cells. Okay. Now, aside from adhesin proteins, they also have Listeria lysine O. The main action of this is that it would lyse, okay, the phagosome membrane. Remember, every time an organism enters human host cells, it will be encased in a bag. No? So it will be encased in a bag. So, it's for phagocytes, we call it the phagosome. Okay, so this would attached to or match with lysosome bags. So pag nagmatch si phagosome and lysosome bags, we have the phagolysosome structure. Okay? So dito pa lang, no, once the organism enters the cell, it will be encased in a phagosome, preparing it for digestion. No? However, because of the Listeriolysin O, the phagosome bag is lysed so that the organism can escape. Ayan. Diba? So internalin A, internalin B for entry, at nakapasok. They're encased in phagosome bags, but because they have Listeria lysine O, they are able to escape. Okay? So once they have escaped, the Act A, okay, Act A or the actin assembly inducing protein will cause polymerization of the actin filaments. Remember, actin filaments are part of the cytoskeleton of host cells, right? So Act A, no, which is another virulence factor of the organism, causes polymerization of the actin filaments. And that would serve as the pivotal elements, pushing the cell outward, no, into the next or the neighboring cell. Ayan. So, tinutulungan ng Act A para mangapit bahay yung bacteria. Okay? So, the Act A pushes, okay, or the Act A polymerizes actin filaments to push out the bacteria, no, from the initially infected cells onto the next target cell. Ayan. So nakapagkapit bahay. Okay? So once inside the neighboring cell, the organism will be encased again in a double layer of membrane. Okay. However, with the aid of lecithinase enzyme, no, and Listeria lysine O plus a phosphatidylcholine specific phospholipase, the organism is still able to escape, no, the two membrane structure. So makakapag-infect ulit siya nung cell, di ba? So again, to recall, virulence factors of Listeria includes adhesin proteins, that will be the fibronectin binding protein A, internalin, of course, internalin A and B, which allows them to attach and penetrate the human cells, and that would be coupled with protein P60, which would increase adhesion and penetration into the mammalian cells. Of course, they have Listeria lysine O, which allows them to escape phagosomes, no, in the initially infected cells, and that would also be responsible for aiding the bacteria to escape the double membrane structure when they invade the neighboring cell. Okay. Act A, which is responsible for the polymerization of actin filaments, pushing the infectious organism out, no, and onto the next neighboring cell. So what are the diseases caused by Listeria monocytogenes? So Listeria is known to cause diseases in pregnant women as well as in neonates. Okay? So in pregnant women, the infection is usually seen during the third trimester. Okay? So it could also lead to spontaneous abortion and stillborn neonates. Okay? In neonates, they cause granulomatous infantiseptica. Okay, so there's neonatal sepsis. In some cases, there are pustular lesions. And then in some, again, there are granulomas found in the organs of the neonate. Okay, that's for the early onset. So as soon as the baby comes out of the infected mother, uh, the early onset of infection is in the form of granulomatous infantiseptica. However, there are some cases where newborns, may come out of their mother's womb looking normal, but after several weeks, okay, they may suffer from the late onset syndrome. Like they have, some neonates may have neonatal meningitis, some may have meningoencephalitis, and then some may have bacteremia. Okay, so early onset means as soon as they are given birth to, nakikita na yung infection. So granulomatous infantiseptica. However, some infants are born looking normal, and then the infection may manifest after a few weeks or months. Okay. So what do we do in the laboratory? So you can culture your organisms on sheep's blood agar, tryptose agar, phenylethylalanine agar, or the McBride agar. Ang McBride agar is actually specific for uh, Listeria monocytogenes. Okay. And then we have nalidixic acid media. Okay. Our organism are capnophiles. They would prefer slightly increased carbon dioxide when cultivated. The growth occurs over a wide range of temperature. They can grow as low as 0.5 degrees Celsius to 45 degrees Celsius. Okay. So we can use the cold enrichment medium. Okay. Because which is usually stored at 4 degrees because our organism is able to survive, cool temperatures. So pwedeng-pwede ang cold enrichment media. Okay, so this is the summary of the assays that we can perform on Listeria monocytogenes. So Listeria is catalase, esculin hydrolysis, and motility, as well as catalase positive, esculin hydrolysis positive. It's motil, it's beta hemolytic, and it's able to grow at 6.5% sodium chloride. Okay, so that's your Listeria. So additional clues, no, to aid you. Your Listeria monocytogenes is able to demonstrate two forms of motility. So when we prepare wet mounts, the organisms are able to demonstrate the tumbling motility. So it appears as if they're rolling on wet mounts. Now, in motility medium, mga semi-solid media, they appear to demonstrate the inverted tree or umbrella structure, gaya niyan. Okay? So wider, okay, the spread is wider at the top and then it thins out towards the bottom. So the same here, eto mukhang payong talaga. Okay? So mukhang umbrella ang itsura. So this one is usually observed when your motility medium is incubated at room temperature between 22 to 25 degrees Celsius, but this does not appear when the culture media is incubated at 35 degrees Celsius or higher. And another is a type of camp test. So the camp test can also be performed on our Listeria monocytogenes. This is actually a test used to demonstrate the ability of Listeria species to cause hemolysis or show hemolytic reactions. So here, a more pronounced camp reaction is seen with Listeria monocytogenes when Rhodococcus equi is placed. Okay, so here we have Rhodococcus equi, and then here we have Staphylococcus aureus, known Staphylococcus. So if you notice, the hemolytic area, okay, of Listeria when matched with S. aureus is really very faint. Pero here with R. equi, you'll see a more clear zone, though konti lang, but still, okay? It's clearer here compared to here. Okay, that's described as a block type of hemolysis. And then we also can go and check for pathogenicity of our Listeria monocytogenes by performing the ocular test of Anton. This is a type of animal inoculation test. So the test animal is rabbit. Okay? So the organisms, maybe a colony or two, the organisms are suspended in sterile water, and then about 2 to 3 drops are placed onto the conjunctival sac of the rabbit's eye. Okay? And then a positive reaction would be purulent conjunctivitis. Ayan, conjunctivitis yung rabbit. So pag nag-conjunctivitis siya, positive yun for a pathogenic or disease-causing Listeria monocytogenes. Okay? So moving on to erysipelathrix ruseopathiae. Medyo marami naman to sila. Kaya lang, again, okay, only ruseopathiae is known to be isolated in human samples. Okay? So the diseases caused by erysipelathrix is erysipeloid. Okay? So it's a local skin infection, no, characterized by ayan, no, parang mga ecchymosis or bruises. Okay? There could be this could be an occupational, an example of an infection acquired because of exposure in the type of work, that the patient does. Usually, yung mga fisherman or yung mga uh, selling vendors, fish vendors, or those that are processing fish for uh, drying, okay, or salting, yeah. O yung nagpipickle ng fish. Okay, so sila yon. Especially if they have cuts on the skin and they get exposed to the organism found in salt water or fish. Okay, so that's erysipeloid. Sometimes erysipelothrics can cause septicemia associated with endocarditis, and in others, they may appear as diffuse cutaneous infections. And so, ito naman yung summary of the characteristics ng ating erysipelothrics. So, your erysipelothrics is negative for catalase production, motility, and esculin hydrolysis, as well as nitrate reduction and urease production. However, they are positive for glucose fermentation and production of acids from there. And they are also able to produce hydrogen sulfide from or on TSI agar. Ayan. So when we do assays of erysipelothrics, one of the most outstanding or remarkable characteristics of the organism is the test tube brush appearance on gelatin media. Ayan, di ba mukhang test tube brush? Ang cute lang. Okay? And then in some cases, they're able to demonstrate their ability to produce hydrogen sulfide. Okay? So this is the only gram-positive bacilli that is able to produce hydrogen sulfide. Okay? And then we have Lactobacillus acidophilus. So this one is actually a very important microbiota in the body. It's also known as the daughter lane bacillus, which can be seen in vaginal smears. Yeah, no? So lactobacillus is found in our mouth, in gastrointestinal tract, in the vaginal tract. They appear to be non-pathogenic, right? But in immune-compromised patient, there may be some sort of bacteremia. Okay, so this one plays a very important role in maintaining an acidic environment in a lot of areas in the body natin. No? So this is because they produce a large amount of lactic acid. Okay? So in the vaginal area, nandun sila. Sa GIT natin, nandun din ang ating mga Lactobacillus acidophilus. Okay? So they grow well on tomato juice agar. Okay? They are gram-positive, non-motile, catalase, hydrogen sulfide, and bile esculin negative. Okay. Then we have Gardnerella. Okay, so Gardnerella vaginalis, which is formerly known as Haemophilus vaginalis. Did you know that previously they were considered as gram-negative organisms? No? Until they did a very detailed study on the cell membrane and the cell wall of Gardnerella. Okay? They realized na wala pala siyang LPS, no? Which is technically found in most, if not all, cell walls of gram-negative organism. Wala siyang lipopolysaccharide. Yun. So, after performing that really detailed study on the cell wall, so gram-positive QDIC ante. Okay, so gram-positive pala talaga si Gardnerella. Okay, so this is known as an agent of vaginosis. Okay, there's no inflammation. Okay, wala namang swelling except that nandun yung organism, and they may cause not really irritation, but observable changes like an increase in discharge. Gaya na nasa picture na nakikita nyo, may ano siya, tawag nito yung a form of vaginitis is different. Okay, so there's vaginitis can be seen in Trichomonas infection. It can be seen in other infections involving the vaginal area. Okay. But for Gardnerella, it's vaginosis. Okay. So just to show you ano yung difference nila. Pag vaginitis, there's redness, there's irritation. In Gardnerella, there's no redness. There's no swelling, but there's increased in discharge and then may foul odor. Yun. Okay? So, our organism is also able to cause neonatal sepsis. Siyempre, kung ipanganak ka ba naman, lalabas ka ba naman sa ganitong itsura, o di, nagkaroon ka talaga ng neonatal sepsis. And then, for females, right, because yung ating, when they give birth, okay, a part of the perineum may tear, especially pag normal delivery. Itong thin membrane na to, napupunit yan. And so, pag may infection, pag merong si mother, may bacterial uh, colonization in the area, tapos may sugat dyan, naku, pwedeng pasukin yun ng organism. Okay? So for identification, we can grow our samples with the suspected organisms on the human blood twin agar, which is the best for our Gardnerella. Okay? So for laboratory, they can perform the sniff test. Ew, aamuyin mo yung discharge. Okay? And then, dadagdagan ng 10% potassium hydroxide. Okay? So fishy, amine, odor ang naaamoy natin. And then, on cytologic evaluation, we'll see clue cells like this. Okay? So clue cells are technically epithelial cells filled with bacteria. Ayan, nagmumukha tulong may glittery ang itsura ng ating squamous epithelial cells. Kato di, bacteria dito. Okay. And then we have nocardia. Okay, so for nocardia, these are unique because these are partially acid fast. Recall our discussion on acid fast. Okay, so apart from Mycobacterium, there are other organisms that would be that is positive for the mycolic acid. So you have nocardia and your corine. Okay, so si corine, very short ang kanyang acid fast or yung kanyang mycolic acids rather. Okay, short. So when we do acid fast staining, negative talaga siya. Si nocardia have slightly longer mycolic acid chains. Okay, slightly longer than corine. Okay, sometimes they may appear weakly acid fast. Okay, or partially acid fast, gaya ni Titch. Ayan. Okay, so to differentiate them from Mycobacteria, si Mycobacteria, meron siya nung coiling, okay? Pero not branching. This one is really branching. Okay? Also, serpentine yung kay Mycobacterium. So, parang mag-chain ng mahaba. Pero, etong kay Nocardia, talagang ganyan, buhol-buhol. Akala mo, fungi. Akala mo, mycelia. Okay? So, they could also grow on SDA. Remember, SDA is a basic culture media. No? Primary culture media for our fungal organisms. Okay? So, they can grow well on SDA provided na wala lang itong wala lang kaning chloramphenicol because they would be killed, or inhibited by the presence of chloramphenicol. Okay, so these are also capnophiles. They would require increase in carbon dioxide about 10%. So they could be incubated for 3 days to up to 4 weeks. Colonies may appear waxy, bumpy, and either slightly yellow, orange, or orange colonies, kagaya ng nasa picture natin. Okay, they are urease positive, no? They're strong urease producers, and they can grow on milk. Okay, if we grow them on milk, that would enhance their acid fastness. Okay, so again, they may resemble Mycobacteria, but remember, kay Mycobacteria parang chains or palisade, pero itong kay Nocardia talaga naman mukha siyang mycelia o yung fungi na nag-grow. Wait lang. Hmm. So they are also strictly aerobic, no, na bacilli. So what are the diseases caused by this organism? So the organism is able to cause, no, they are able to enter the body rather by via inhalation. So yung mga bacilli na nasa contaminated airborne droplet, pag na-inhale natin yon, so the organism can enter the body. Okay? The infection may begin as chronic lobar pneumonia, which would mimic TB sometimes, but pwede naman talagang mag-disseminate. So look at this, for example, no? So, grabe ka-hazy ng kanyang lungs na puno ng lesion all over, no? Yung mga microcolonies of the organism. So, fatal pulmonary nocardiosis. Or, pag disseminated naman, ayan, meron din namang involvement of the brain tissue. So if you notice, ayan, nagkakamatayan ang mga neurons on this part, kaya blanco. Okay? So the lesions are caused by nocardia asteroides, no, a species of nocardia. May ganun. Also, nocardia is a part of the actinomycetes group that can cause the actinomycetoma or yung bacterial type of mycetoma, no, or madura foot. So it's a type of infection that's shared by fungi and bacteria. So both fungi and bacteria can cause, no, actinomycetoma. Okay? So there's localized low progressing, no, chronic infection, as in parang malata ito, nagkakaroon ng mga draining sinuses. So it's destructive but painless. Okay? So usually the reason why mabilis mag-progress ang infection like this, no, maabot na dyan. Usually dito lang naman start yan sa paa. Okay? And then umaabot hanggang dun sa taas. Gumagrabe ng ganito basically because of this, no, painless, di bayat na magpa-doktor dayon kung may sakit, di ba? So pinapabayaan lang natin dahil wala namang sakit, pero tissues are destroyed in the process. Okay? So the longer you delay treatment, the more tissues are destroyed in the process. Okay, so this is caused by, again, actinomycetes, which would include nocardia, actinomyces, yan, no? Sila yun, yung nagkakos ng actinomycetoma or fungi. Okay, so both nocardia asteroides and nocardia brasiliensis can cause actinomycetoma under nocardia group. Okay, so moving on with our actinomyces. So another actinomycete that can cause mycetoma also. So they're non-acid fast. They would look like nocardia, but they're non-acid fast. They're facultative anaerobes. They may be found in rod shapes, coccoid, or coriniform like this. No mycelia, unlike in nocardia. Okay? So, sinocardia grabe branch ang OA. Ito pwedeng may short branches. Okay? So, the most important species is Actinomyces israeli. And then we have a recent, no, na addition. We have the Actinomyces gerenxeriae. Diba? So, actinomycosis, no? Or actinomycetoma, no? Pag actinomycetoma, yun yun, no? Yung madura foot. Okay? So, actinomycosis, chronic infection, okay? Suppurative, meron tayong mga sinuses, no? Na draining sinuses, okay? Pwedeng cervicofacial, thoracic, and abdominal. Okay? So, actinomycosis, eto yun. Actinomycetoma, yung isa, no? Pero pwedeng-pwede rin naman itong mag-cause na actinomycetoma. Okay. So laboratory diagnosis, you can work with pus, sputum, tissue samples. Okay. We can culture them on brain heart infusion and thioglycolate at 37 degrees Celsius. Filaments may break into short chains. Okay. Remember, sinocardia yung mas grabe mag-branching. Siya, short chains lang. Okay. And then some may show coccobacillary forms. Now, usually, okay, pag early, okay, almost say 24 hours incubation, they would produce the usual colonies, convex, yeah, no, round. But as they age, okay, after 48 hours of incubation, nagko-collapse yung center, creating parang a crater at the middle. Okay? And then the edges would uh, fall down. Okay? So giving the characteristic molar tooth colonies. Ayan, mukhang molar tooth, di ba? Dahil sa crater sa gitna. Okay, so molar tooth colonies and ano yung isa nating na ano ngayon? Test tube brush colonies. And then may umbrella motility. O yan. So tandaan yung mga descriptions na ganyan. Okay. So this one, no, infection involving actinomyces may be treated with penicillin, okay, or erythromycin, clindamycin, and syempre, surgical excision and drainage of the pus. And then we have the last member or the last group under gram-positive bacilli, we have Propionibacterium. Okay, so isali natin to dahil marami sa atin ang nabiktima ng organism na to. Okay, so Propionibacterium is a facultative anaerobic organism similar to your actinomyces. They're non-spore-forming, non-motile, irregular rods, or sometimes they appear as short-branched rods. No, temperature for incubation is best at 30 degrees Celsius. Normal flora ito ng ating skin, no, ng oral cavity, ng GIT, and even the ears. They are able to metabolize carbohydrates and lactate, no. They convert lactic acid to propionic acid and carbon dioxide. Okay. The most important species for this member is Propionibacterium acnes. No? So this one is considered opportunistic. Okay? They're implicated in acne, okay? Acne vulgaris, sometimes endocarditis or shunt infections, no? Yung mga sa mga patients sa ICU, okay? Ginagawan ng shunt para mas madaling makakuha ng blood sample or maka-introduce ng gamot, no, sa kanyang vein. So ginagawan ng shunt or parang sinusugatan. Okay? And then, what else? They are able, so their virulence factor is their ability to produce lipase. Okay, so lipase is an enzyme that can split free fatty acid from skin lipids, causing irritation. No? So kaya nagkakaroon tayo ng mga acne. Ito ang dahilan ng ating pimples. Ayan, hindi ba? So we culture them anaerobically on blood agar plate. No, they grow really small colonies. No, around 1 to 2 millimeters in diameter, circular and prior, convex, glistening, and opaque. Okay, some may produce hemolysis. Okay? Ayan, no. So pag immunofluorescence, ganyan. Or when you culture them. Sometimes they produce narrow zone of hemolysis. Ayan, no? So, alam niyo naman, ito yung mga pores natin sa ating mukha. Okay? So, pag nakapasok yung organism doon, it can block, no? It can cause accumulation of dirt. Okay? Nagba-block siya dyan. So, mag-accumulate yung dirt, yung oil, causing your the causing sebum to uh, increase, no, dun sa area na yon, sa maliit na area na yan. So nagkaka-inflame, no? Ayan. Oh, so naba-block yung daanan kasi may dirt, nandun na din yung bacteria. So walang lalabasan yung oil. Kaya they get stuck up here. And so they cause irritation, swelling. Ayan, no? Nagkaroon na ng pustule formation dahil hindi nga makalabas. So ayan, nabulok na dun sa ating mga pores. Okay? So ito yung mga nakukos niya, di ba? Acne fulminans, tsaka acne vulgaris naman. So this can be treated by really strong drugs like isotretinoin, Accutane. Grabe ang treatment, no, pag ganito. So, pero yung ma-stop, no, the infection may be stopped, pero the pits will stay. So, nagko-cause din siya ng, tawag doon, ng depression, no? Even the drugs given would cause, no, it has an effect sa immune system natin. Okay? Grabe, di ba? So, pwede naman siyang ipa-derma, pero napakamahal ng treatment na 'to. Okay, so kailangan siyang magpa-diamond peel na bonggang-bongga, and then mag-Spanish peel para ma-lessen yung pagkalalim ng kanyang pimple or yung kanyang pits. So, first thing is really to stop, no, infection. Sa kalapad ng skin diri jud sila motubo, no? Kasi nandun naman yung maraming oil glands. Okay? Sometimes ganyan sa back. Grabe. Okay? So acne vulgaris. Bulgar na bulgar ang kanyang acne. Okay? So that's it, no, for our non-spore forming gram-positive bacilli. Thank you for watching.